Director of the Environmental and Health over the Lifecourse program at ISGlobal here in Barcelona. Her research is driven by the need to protect vulnerable populations and in particular children, and to understand the harmful effects of environmental exposures early in life. She's pioneered and led many European projects, the Helix project, the H2020 Athlete projects, and iHunt, among others, and part of her work has been to create or coordinate projects that set up an infrastructure in Europe to get at and having a bigger database of the exposome, with the ultimate goal to understand how the exposome influences early in life health in children. So she spearheaded the study of the exposome and multi-omics determinants of child health. And thank you so much for being here, Martine. I'm really looking forward to hear your talk.
Good. Thank you, Marta, for the invitation. It's a real pleasure to be here. It's my first time at the BSc, and so I want to talk about going beyond the genome, and I will zoom in on the environment, especially on the exposome. So I'm going to give you some backgrounds on what the exposome is, how it was conceptualized, and then we're going to look at some implementations in our own studies, focusing on the early part of the life course. I don't know how to go forward here. Oh, I have to. Sorry. Okay. No.
Yeah, there you
go. Okay, so Marta has already given the introduction. We have several big research programs at ISGlobal, and I lead one of them, the Environment and Health over the Life course program. And our overall aim is to look at the causes and mechanisms of non-communicable diseases, especially the environmental, occupational, lifestyle, social, and genetic risk factors throughout the life course. So this is very broad, and it goes all the way from prenatal to later adult life. And within that, my own lines of research focus on children and environment. So we look at a whole range of different environmental exposures, biological responses, and different health outcomes that we measure in children, including neurodevelopment, cardiometabolic health, respiratory health, fetal growth and birth outcomes, and also puberty-related outcomes. So as I said, I want to give you some introduction to environmental exposures and the exposome, and then show you some implementation, some examples of how we do exposome studies focused on our early life research. So in terms of the big picture and environmental risk factors for disease, there are some estimates showing that about 24% of all global deaths, global mortality, is linked to the environment, to environmental exposures. So it's not a negligible part of our disease causation. And that includes a very wide range of factors. Air pollution, both indoor and outdoor, chemical pollutants. Here they also include water sanitation, occupational risks, but also physical exposures like noise or temperature, and our surroundings, our built environments, and how they influence our disease risks. So this is a very wide and complex picture. The environment is not one simple exposure. And here's another estimate, which is similar, a little bit lower here. This is the global burden of disease exercise, and they estimate that pollution is the largest environmental cause of disease and premature death today in the world. And diseases by pollution in this exercise were responsible for about 16% of all the deaths worldwide. And that was more than AIDS, tuberculosis, and malaria combined. So this is a substantial proportion of disease. Now, there's some notes here to take, and that is, first of all, that most of this burden that was calculated here, estimated here, 70% was caused by air pollution mixtures. Yeah, so both indoor and outdoor air pollution. So that is by far the largest contributor in this exercise. And then I think it's really important to recognize that many environmental risks are not included in this type of calculations because we don't have good enough data. To do these calculations, you need data on the dose response, so on how the exposure correlates with the disease outcome, and you need good data on exposure globally. And that is not available for many of the environmental exposures that we might think of. For example, chemical pollution, the effects are really poorly defined. We don't have good risk estimates. So it's almost certainly underestimated, this environmental burden of disease. So we feel, in fact, it could be a lot higher than that. So we're dealing with a large problem, but one that is, that for the large part, we don't actually know very much about. And the one picture that shows that very clearly is this one, which is the iceberg of the chemical pollution problem. There's about 100,000 manufactured chemicals on the market, but health risk data or risk assessment only exists for a small fraction of that. Maybe 500 chemicals are at the tip of the iceberg, but the rest is all underwater because there's just not a good enough risk assessment data. And several thousand of these chemicals are actually causing very widespread pollution exposure in the population. So they're everyday chemicals that are used in many products and that really are widespread in the population. So there is a problem here because there's so much that we don't know about this whole chemical soup. And then there are many new concerns that we know even less about. I'm put here a slide on micro and nanoplastics because the whole plastics problem is also associated, of course, with the chemical problem, with many plastics-related chemicals that, for example, are listed down there, that really are causing a large problem, both the plastic particles and the chemicals that are adhered to these plastics. But we have very little data on the health risks. So there's many environmental problems that we haven't really begun to tackle. And on the other hand, we also know that disease is not only caused by genetics. If we look, for example, here in this figure, there's the heritable contribution for a selected group of communicable diseases, non-communicable diseases. And we see that the genetic part calculated there's a wide range, obviously, but it goes at maximum there to 48% for asthma risk. So there's a large part of these type of chronic diseases, including type 2 diabetes and cancers, asthma, etc., that are not explained by the genetic risks. So there is a large environmental component. But then if we look at the right, and this is the the proportion of the burden of disease attributed to environmental, occupational, and metabolic risk factors, we see that at most about 50% is explained by all the factors that we do know about. So there's this whole missing part in disease causation that is due to environment, not necessarily environmental pollution, but many environmental factors. It could be social, lifestyle, or much broader environment that we don't know much about these causes. So this is a little bit how the exposome was born. No? The idea 20 years ago, Chris Wild, who was the director of IAC, of the International Agency for Research of Cancer, he said, well, a lot of investment has gone into characterizing our genome, huge investment, tools, etc. And we know a lot about the genome. But on the other hand, we have this whole environmental burden of disease that is far less well characterized. So there's this disparity. And he proposed the word exposome to draw attention to that disparity and to the need to invest more in into this environmental part of the balance. And so it was really a way to draw attention to the fact that we don't know enough about our environmental causes of disease. So the exposome here was defined as representing all the environmental exposures, including diet, lifestyle, etc., from conception onwards, as a quantity of critical interest in disease etiology. So a lot of the inspiration is in the exposome field is drawn from genomics. You know, there's a parallel here. The complete set of environmental exposures over a lifetime versus the complete set of genes in a particular organism. And with the idea that if we can well characterize the two, then we can hopefully get much better information on disease causation. So that was 20 years ago that this concept was proposed. And there's a good commentary actually for anyone who's interested on a little bit on the historical perspective, also written by Chris Wilde. And he said after 20 years, what we do notice that the exposome is hugely complex. No, it's not like taking one drop of blood and measuring your genome. It's over a lifetime, many different variables. And it's practically unmeasurable in its entirety. But that's what we do know is that we cannot measure everybody's exposome because it's over the whole lifetime, because it's dynamic, it changes all the time, and there is a huge number of possible factors that could play a role, and it's incredibly complex. So it's practically unmeasurable. So therefore, we have to start with partial characterizations. So even with partial characterization, so defining or measuring a part of the exposome, that would offer major opportunities to understand disease etiology, including through exposome studies applied to large prospective cohorts, where we follow populations over time. And he also clearly said the way forward is not to argue whether a study of genes or environment is more important, but we have to see both as necessary and complementary. So that's all fairly conceptual, but we can look at some of the characteristics of the exposome that can help us to move forward with these more complex questions, no, for environment and health. Because up to now, most studies in this field focus on one exposure and one health outcome. No? So we've been thinking carefully about how we can use the exposome as a framework to make progress on the more complex questions. So really going into the complexities. And that would first be, using this comprehensive characteristic of the exposome to move towards better integration of different types of exposures, including the chemical, physical, biological, and psychosocial stressors. So to really try and integrate and combine exposures from these different domains into our studies. And that would allow us then to look at combinations of exposures, interactions, mixtures. So we're not just doing every study on one exposure and one health outcome. Then, of course, there's the Lifecourse characteristic. This is very much a Lifecourse concept, whereby the temporal sequences across the Lifecourse are important. And also this allows us to look at vulnerable time periods. If you have data on different points in life, you may be able to target certain vulnerable periods. There's a lot of emphasis in the exposome fields on developing exposure assessment tools that can measure this wider range of environmental factors. So moving towards tools that can measure multiple environmental exposures. And here there's a very wide range of different tools available. For example, we can use satellite data to map our outdoor environment. So there's a lot of work being done on geospatial methods, mapping for example, our built environment exposures. We can do a lot of determinations of neighborhood environments just by looking at the maps. We can see how many green spaces, how many parks there are. We can look at air pollution maps, noise maps, maps of light, etc. And this is a powerful tool for the outdoor environment. At a more personal level, we can give people wearables, sensors to wear that can measure their exposures in a much more personal level. And then on the chemical side, there's a lot of movement now to developing the metabolomics platforms that already exist, but to develop those to also measure exogenous chemical exposures. That means not just determining the metabolites that are endogenous, but also using the same platforms to go to the exogenous molecules. And there's good developments there to allow us to measure many, many, many more chemicals in a biological sample. Then there's the integration of biology. So the exposome has a strong focus also on trying to link exposure, biology, and disease. So that is to look for markers that are markers of early biological responses that could help us to develop early biomarkers or improve our mechanistic understanding of some of the effects between the environment and the disease. And then, because it's an ohm, the exposome is also has a discovery-driven characteristic. So it kind of moves away a bit from the very focused hypothesis-driven research to discovery-driven, allowing sort of for new and unanticipated associations to emerge, paralleling the genome-wide association studies. So where can this help? And as I said, this is more really to go into the complexities. We can systematically analyze many exposures. We can look at complex mixtures, interactions, integrate the biology, and use some of these methods to see if we can discover new exposures that have not yet been measured before. So I'm going to move now to some of the studies we've been doing in early life to try and see if we can implement some of these concepts, because it's a complex concept. And when we first started working on this, we had no idea what we were really starting to work with. So why early life? These are key stages of vulnerability. And they're also key stages of life for implementing cost-effective prevention actions. So if we can intervene on our environmental risk factors in early life, before the disease occurs, we can implement much more effective and efficient intervention. Well, that means preventing before somebody gets ill. So that's why early life could actually be a very good point of intervention. And why are these periods vulnerable? First of all, because there's very rapid organ development during the early years of life. During pregnancy, infancy, childhood, and adolescence, there are very specific and very distinct periods of development. And when an organ is developing rapidly, any environmental insult could have a more pronounced effect. We know that certain chronic diseases have at least part of their origin in early life. So we know that children who have certain, for example, children who are obese have a much higher risk of developing later cardiovascular disease, or children who have neurodevelopmental disadvantages have a much higher risk of later on developing mental health problems. So there are specific chronic diseases that are clearly linked to what happens in early life. Anything that happens in early life has a lifetime influence, as I said already, and this is an important point for prevention. So here are two examples of diseases that seem to have at least part of their origin in early life. Well, obesity is not a disease, of course, but we do know that obese children are much more likely to become diseased adults with all the risks that involves for chronic disease. And on the right-hand side, these are lung function trajectories. And again, we see that certain trajectories of lung function in early life are clearly linked to later lung function. So there's this build-up part in early life that if you don't reach the peak lung function capacity by the time you're 20, then you're much more likely to suffer later on from diseases like COPD. So we have some clear examples of diseases that are originating in early life. So how do we study this? And this is where I'm an environmental epidemiologist, so we run epidemiological studies, observational studies. These are not experiments. We observe the general population in these studies. What we do is we recruit pregnant women into our studies. We take biological samples in different trimesters of pregnancy and at birth, and we ask the mothers a lot of questions. So they get long questionnaires on diet, on social factors, on all their lifestyle factors, and we ask them where they live, so we can pinpoint their residential addresses, and then we follow the children as they grow up. So in our studies, the children, we have visits, examinations of the children every 2 or 3 years, in infancy, childhood, and now all the way to adolescence and hopefully further on. And then we measure many exposures using some of the methods I just mentioned, the geospatial methods for the outdoor environment, we measure chemical biomarkers using chemical analysis labs. And then in our exposome studies, we also integrate different omics layers. We've measured the genetics of the children, and then we do health examinations. So we measure their cardiometabolic health, their neurodevelopment. So they undergo a battery of tests and questionnaires, and respiratory health. We test their lung function, and we have questionnaires. So that's where a lot of our studies are based on these observational studies. This is an example of the athlete project, which is a large European project that we coordinated on the exposome in early life, where we also try to harmonize a lot of these type of data sets from different European countries. So the one example I wanted to show you here is the Helix cohort that we started building in one of the earlier European projects quite some years ago. I think we started Helix in 2013. And this is just an example of what an exposome database could look like. Obviously, this can be added to and expanded. We determined the exposome measuring a very long list of exposures during pregnancy and again in childhood. And now we've just finished also the follow-up in adolescence with the same groups of exposures. So we have now in this cohort 3 time points for the exposures. And then we measured different omics layers. Now this is work that started in 2013. So some of these methods are now not so new anymore, but at the time it was the best available for the resources we had. So here we had the DNA methylation expression with expression arrays. In the children here we have plasma proteins, but this was very targeted work, and serum and urine metabolites. We've now since added other layers of omics with more proteomics, etc. And we've done a follow-up, as I said, in adolescence, where we also added new omics layers. We have now metagenomics data using from the stool samples of the adolescents and proteomics, metabolomics, etc. So this is being added onto, but it gives us a complex database to work with. And this database includes about 1,600 mother-child pairs. About 850 participated in the adolescent follow-up. And then one of our first questions was, so how do you link this complex picture, not as complex exposures, to the health outcomes in the children? And that's not so easy, because these are incredibly highly correlated exposures. This is a picture of the hundreds or so exposures that we measured in the children. And this is the correlation network. So you can see that there's many correlations. They're stronger within families of exposures, but there's still a lot of correlation also between families of exposure. So we looked at different methods for linking that to child health outcomes. And there's different ways of doing that. You can run an exposome-wide analysis, very similar to a GWAT, where you link every exposure with the outcome. And then you can go further, apply variable selection methods. You can look at people who share similar exposomes, more cluster analyses, for example. You can combine exposure variables, reducing the dimension of that picture, which is not so easy. We tried some dimension reduction on this whole matrix, and you don't get very good principal components. Yeah, so that's that wasn't so easy. And now we're also looking at, we've applied several of these methods that can use, they build mixtures of multiple correlated exposures. I'll give some examples of that. But there's methods available that allow you to build a mixture index and looking at the exposures that relate, combined exposures relating to an outcome. So these are some of the statistical techniques that could be used for different, obviously they answer different questions. So I want to show you some examples. This is one study we did on how the exposome relates to child blood pressure. Now, blood pressure in children is already an important marker of later cardiovascular health. So this is these are healthy children, but we can still, the distribution of blood pressure in children already tells us something about later disease. So first stop is an Expos, where we found several of these environmental exposures linked to the blood pressure in children, but in both directions. Some of the exposures seem to be linked to lower, others to higher blood pressure, but at least it gave us a systematic way to look at all these factors together, which has not been done before. Not a study on air pollution and blood pressure, maybe in children, but there was no complete picture. So this is kind of a first way of systematically showing all these associations. And then we follow this with this is one of the variable selection methods, deletion, substitution, addition algorithm that pulls out the set of variables that best predict the outcome. And we got a set of exposures that were related to blood pressure in the children, both during pregnancy and in childhood. I won't go in too deeply on the results here. And the second example is a way to look at mixtures. In this case, we looked at mixtures of endocrine disrupting chemicals, and we were interested in the metabolic syndrome risk in children. So children don't really have a metabolic syndrome as such, but you can already look at the indicators of metabolic risk. So we created an index, which is actually validated in other studies based on waist circumference, blood pressure, insulin, etc. at 8 years of age. And we had measurements of, I think, around 40 chemicals that were measured in the blood and the urine samples of the mothers. So these are concentrations measured in the mothers during the pregnancy. And the novelty about this is that instead of looking at each chemical separately, we combine them. So when you look at them all separately, you don't see such big increases in risk, or hardly any. When you look at the mixtures, you see much clearer results related to this metabolic index. for example, the metals, the group of metals, which includes lead and cadmium and mercury, these heavy metals that we know are toxic. But here they were clearly associated with the metabolic syndrome risk of the children. So this is the mother's exposure to these chemicals linked to the 8-year-old children's metabolic risk. And it's the same with some of these persistent organic pesticides. With the PFAS, you might have heard of PFAS. These are the perfluorinated compounds that we call these forever chemicals that are a lot in the news right now. And there's some pretty good evidence also in toxicology studies that these PFAS do have an effect on lipid metabolism, etc. So this kind of matches with what we see in the toxicology literature. Some other groups of chemicals seem to have some reverse effects that are much harder to explain. But anyway, this is an example of how you can use exposome data to get better information on mixtures of exposures. And then this method also gives us some ideas of the chemicals that most contribute to these mixtures. My third example is on clustering. So this is a clustering technique, the hierarchical clusters that we use to group together exposures in the outdoor environment. So here we had a larger group of children, about 4,000 or 5,000, with fully documented exposures in their external environment, including a lot of variables related to air pollution, traffic, noise, green spaces, building density, walkability, those type of variables. So we had different clusters. 2 of those seem to be the more hazardous clusters, so that with the higher exposures to air pollution and traffic and noise, and lower in green spaces, and those were associated with, in this case, higher diastolic blood pressure, and in very young children, in 4 to 5-year-old children. So that's another way to start looking at exposure data in combined way, looking at profiles, clusters of people with similar exposomes. So doing that, we ran a series of studies on different health endpoints. And we started to get a picture by doing this systematically of which exposures might be more important for certain outcomes. You see a whole list for blood pressure there. This is with pregnancy exposures. We did similar work with liver injury markers, so enzymes, liver enzymes. And then also for childhood, we got a slightly different picture, but this is just to show you how you can use exposome data to get some more systematic and comprehensive ideas of which risk factors may be important for certain, in this case, child health outcomes. So our next question then was, here we do the exposome with each health outcome separately, but from a public health point of view, it's also important to know if there are exposures that are really linked to more than one unhealthy outcome, because those would be the ones that you would want to prioritize. If there's really factors in the environment that link to multiple health outcomes. So we started working on outcome-wide exposome modeling. This is done through reduced rank regression modeling. I'm not an expert on that, but my colleagues have been developing these methods, and the idea is that we can, this uses latent factors to link many of exposures with many outcomes. And in this case, we had 4 outcome domains: cardiometabolic outcomes, mental outcomes, respiratory outcomes, and allergy-related outcomes. And then we also had an overall health outcome score. Now, the idea is that by doing this, you could try to identify what they call here the master regulators of health, so predictors with cross-domain health impact. And that, as I said, is of interest for public health and prevention. We also hope, using this method, we can try to reduce the multiple testing errors And it increases the ability to detect weak signals. And there's some methodological work behind this, which has already been published, and the paper itself is currently under review. So this is kind of, I won't go into every result here, but then you get a very interesting, complete picture of all the associations, and the ones that came out as being important were, again, maternal smoking, being related to many of the different health endpoints. Indoor air pollution, so we had models of indoor air pollution that were consistently associated with more than one health outcome. Some of the endocrine disrupting chemicals, some diet factors, some neighborhood factors. And we also saw that some of these effects persisted into adolescence. So this is a very much a good example of an exposome approach, that we're moving away from this one exposure, one health endpoint. And then the other parts we've been working on is the integration of these biological responses. So here our main interest is in seeing if we can identify molecular omics signatures associated with the environmental exposure. And this is of interest for us, firstly, to see if we can improve our understanding of the biological mechanisms and thereby improve some of the causal inference, biological plausibility. Some of the markers may also be interesting in terms of predicting past exposures. And there's some studies on smoking, for example, that we use where you can use the omics markers to look at what somebody's past exposure was in a better way than asking them in a questionnaire. And also predicting future health outcomes. With potential applications in public health, like if we can detect early subtle effects before a disease occurs, there might be more potential there for prevention, and then possibly in some way in more targeted prevention if we can reverse the changes through interventions. So the first thing we did in the Helix database was to build an atlas of the molecular, the multi-omic signatures related to all the exposures that we measured. So this was in the children, the same exposures I showed you earlier with all the omic signatures that we had available at that time, through an exposome omics-wide association study, and publishing the results in an association atlas. And that gave us networks of exposures and omic signatures. And this is really this is one by one. This is running all the separate regression models. In the pregnancy, we see some fairly disconnected network with a few exposures that seem to cluster around several omic signatures. For example, tobacco smoke again, molybdenum, which is a sexual metal, and some of the endocrine disruptors, giving us some signals in the omics layers in the children. And then in the childhood, so these are childhood exposures linked to the children's omics signatures, we see much more connected networks. And we also see different signatures coming out there. Some of them around the chemicals, but also some around the meteorological factors that we measured, the indoor air pollution, and some dietary factors as well. So you can also, of course, get signals, especially in the metabolomics layers. You see imprints of the diet. So here, I can't go through all the examples. What we also saw is that in the pregnancy, when we look at the pregnancy exposures, the imprints we see in children are mainly, the majority is in the epigenome as a much more long-term imprint in the children. So this is cross-sectionally, we see it is much more spread around all the different omics layers. I can't go through each of all of the results, but these are some of the sort of more pronounced signatures. The maternal smoking showed a robust association with long-lasting imprints in the child methylome. And this was also followed up with another study that looked at the dose responses with quite clear signals that also match with what other studies have found. And similarly for indoor air pollution, we see interesting signals, especially in the metabolites, in the metabolome. So we can start to zoom in on some of these signatures. Other work that can be done is because that first work doesn't include any of the health outcomes. So in some of our next papers, we've looked at some of these signals relating the exposure and the health endpoint. So more maybe what we call a meet in the middle approach, where you look at signals that are overlapped on both sides. And for example, this is with our liver injury markers. So these are liver enzymes that we measured that indicate, again, a higher risk of liver injury. And we find some profiles, inflammatory cytokines, for example, related to mercury exposure that also relate to the liver injury. And the PFAS, again, that are clearly linked to the liver injury risk, and with certain signatures in the metabolome, with certain pathways. So there's also those type of associations that we can find. Then we've also used these data to try and build phenotypic clusters. So we think of childhood obesity. This is a very diverse phenotype. So what we've tried to do is use the multi-omics data to build clusters within the obesity phenotype to look at some more metabolically healthy and unhealthy clusters. And this is work that was published last year, which allowed us to distinguish 3 different clusters. And one of those is the high risk one, which was characterized by higher adiposity, but also a higher degree of metabolic complications. And we have the omics network that's behind that phenotype. And then we link that to our exposome. So we found, again, some environmental pollutants, some of the PFAS and mercury, again, to identify those as important determinants of this high risk cluster. So those are a few examples. We're working on other ways of integrating that. We've done a first work on seeing if we can build exposome risk scores. So this is trying to see, with all the variables in the database, if we can predict health outcomes. And this was done for mental health, for the metabolic risk, and for lung function. And we see different feature importance depending on the type of health outcome. So for mental health, you get quite a different set of predictors than, for example, for metabolic health. So for metabolic health, the metabolome features were really important. Whereas for mental health, there were also variables like maternal stress that were important. So this is starting to sort of put together a predictive scores. I have to admit that this is still in the cross-sectional data set, so this needs to be done prospectively. But it's a first work towards developing also some scores that might be more predictive scores. So there's many challenges in these data sets. We're working with high dimensionality, large numbers of exposures, limited sample size. So doing this work in 1,000 or 1,500 children is not a large data set. And that especially when you work with environmental exposures, they don't have such high risk, relative risks. No? So if we think of smoking and lung cancer, you get high relative risks. When you look at air pollution or one of the chemicals, your risk increases are small. So we're dealing with sort of low-level chronic and mixed exposures. So the associations are weak often. And with limited sample sizes, it's hard to get strong effect sizes. So this should really be done in much larger databases. And then we're also dealing with the multicollinearity. So strong correlations among the exposures make it difficult sometimes to know which is the exposure that is really responsible for the effect that you see. So there's a list of some other challenges that we've encountered: unstable estimates, loss of power, false positives, false negatives, etc. So here's some ideas of how that we might move forward with that. In terms of causality, I think that's our most important challenge in observational studies. These are not experiments. We can't do randomized trials with environmental exposures, so we're kind of observing, but we can't control everything. So one way is to try to integrate all this work much more with experimental models. And there is a nice example in the literature. This is a paper in Science a few years back where they did very similar work to what we did. They created these mixtures of chemicals using this weighted quantile sum regression. So they had a mixture that was related to, in this case, neurodevelopment again in children. So they had a mixture of chemicals that was together, these combination of chemicals that was related to neurodevelopment scores in the children. And they created that mixture in the lab and used experimental systems to test that mixture. And they found very similar, they found certain effects that mimicked what was found in the epidemiological study, in the zebrafish and in the in vitro system. With very similar approaches. So there are, obviously, it's never easy to translate what happens in the zebrafish to a neurodevelopment score in a child, but still there is an opportunity here to really match what happens in the cohorts to what happens on a molecular and experimental level. So that's one example, I think, of how this could move forward. Another part that we are working on is really to replicate in larger databases and larger trying to build harmonized databases with other cohorts. So that's another part that we've been working on in several European projects, is to get harmonized databases that we analyze through federated data infrastructures and really replicating. This is still in observational studies, but for some exposures, it's not that easy to find good model systems. So we're still stuck with the observational studies. And here, I think it's really important to build these larger infrastructures. It's hard to get databases that are really comparable. So for this type of work, but for some exposures, it works much better than others. So we've done work, for example, across all the European cohorts in looking at air pollution effects, and those type of studies are very feasible. When it comes to measuring all these chemical exposures, it's much harder to get comparable data. And then third, I thought, because I'm here, maybe I should talk about AI, which I'm really don't have any expertise in, but I know that there's people in the exposome field who are looking into how we can use the generative AI and the LLMs to guide also some of the exposomics work. And really, most of this work, I think, is looking at how you can pull in all the data that is available in libraries, etc., to get more biologically grounded representations from these data sets. So in using these models to integrate the biological knowledge. So there are some first papers appearing on this. And again, I'm not an expert, but I have some colleagues who are looking into this and seeing how we can start using the AI to, for data augmentation and to have to better integrate the biological knowledge. So thank you very much. This work was done by many people across several European projects. So there's too many to name by name, but I have to acknowledge everybody who worked in this project. I also want to specifically thank some of my colleagues at ISGlobal who've done most of the work on the papers that I showed you. And that's Lea Metre, Augusto Anguita, and Mariona Bustamante. So if any of you are interested in learning more, then please come and talk to us. I will stop there. I don't know how I'm doing for time.
Thanks, question.
Thanks a lot for the talk. It was very inspiring. I would have a question on the data acquisition. You showed the data acquisition, so you're using not questionnaires. So have you tried in some of these projects to directly extract information from clinical records or like unstructured data to like syndromic information or social determinants of health or other kinds of?
We haven't tried to do that. It's true that all of our data comes from the questionnaires that were filled in by the mothers, and now also now that we are dealing with adolescents, so we have empty filling questionnaires, but in some of our cohorts we've gone back to hospital records, but to get some information maybe on the prenatal records, for example, to look at ultrasound data of the baby. So we've gone back to records, but we haven't used any systematic approach for that at all. But that's a nice idea.
We work on like data extraction from unstructured like clinical records, field reports, and it's very interesting to see.
So these are there's a lot more possible now than when you have to think these cohorts started more than 20 years ago. So that a lot of what was done then was done by hand or by not even all the data was digitalized at that point. We had someone go to the records and write down the baby's weight and height and things like that, which is, if we were setting up a new cohort now, we could find much better ways to do that. Good.
So Claudia, go ahead.
Yeah. Thank you for your talk. It's super interesting, this topic. I wasn't aware about the exposome, so for me it's like completely wide eye-opening. Till now you are dealing with a lot of variables, and I will like to pinch you with one more variable now that you're reaching to the teenagers. That's the sex or gender differences, how they may affect even the study from beyond. That's only for picking your brain.
I mean, it's a very good topic. In this study, we had, I think, one question somewhere in the questionnaire about gender identity, etc. And it's not a very large study, so we don't have many of the divergent identities, but there's a few, and we haven't really done much with that data yet. Very nice. Yeah.
Yes, so thanks for the talk. It was great. My question is about diet.
Yeah.
Especially throughout the presentation, it seems like it doesn't affect that much the genetics or DNA methylation or most of the omics. But then when you were comparing pregnancy effects and children effect, it was very clear impacting children development, but not the pregnancy, where evolution and more have an effect. Do you think that didn't show
First of all, is measurement error. I think we didn't always it's hard to measure diet accurately. So and here in the pregnancy questionnaires, as I said, this was done 20 years ago, and there's I think quite a lot of measurement error in the data. And I think we try to do a better job in the children. You always get errors. So it's not always easy. We're trying to do some more work now with creating sort of diet profiles, and maybe that works better than asking for looking at specific food groups, because it's hard to see an effect of one particular food group when you use these type of questionnaires. I think this wasn't a diet study, so it wasn't the biggest focus of the study. And then I think in the omics layers, the profiles we see related to diet are mainly the metabolomic. A lot of it is the metabolomics. It's probably what you expect. And there's good databases also linking diet sources to metabolic features, metabolomics features. So I think it's maybe a more short-term profile. So whatever the mother ate in the pregnancy is not going to influence the child's metabolic profile. It's more that metabolic profile is more linked to what the child ate rather than what the mother ate. So, yeah, there are larger maybe epigenetic studies ongoing. So that's maybe where we might see more permanent signatures.
Next question.
My question is related to some of the challenges you mentioned. I would assume, or I think that probably the child and the mother exposomes are highly correlated, right? So how do you disentangle this? Does your data set allow you to already rule out correlations, or you do this methodologically to assert if one outcome is due to the prenatal or the child?
Yeah, there are correlations, and I didn't show you. We did look at that in our early papers. We looked at the correlations between the children and the mothers, and it depends which family of exposures you look at. Some variables are very highly correlated, and others much less so. So, for example, if you live on the same address since your pregnancy, your evolution exposure is highly correlated each year of life. It doesn't change that much. There's a little bit of a trend, for example, here in Barcelona, we're declining a little bit of air pollution, but it's super highly correlated. So the only real changes you see is when people move. But for others, for some of the endocrine disrupting chemicals, for example, some of those are very non-persistent, so they're highly variable, and they don't actually correlate at all between the mother and the child. So your exposure to whatever these plastics, chemicals, these phthalates, for example, is not really related to what your mother was exposed to, because it's related to maybe the cosmetics products they use and certain plastics packaging. And so it really depends on the sources. What we do if there's a high correlation is we try to adjust. So the models in childhood are adjusted for the pregnancy exposures that were highly associated with the same outcome. So you try to take out the confounding effect. It may be, this is another option for the future, maybe, is to build models that look more at cumulative exposures. It's quite tricky, but it can be done maybe for some of them. Yeah, but your repeated exposures is it's a real challenge in these data sets. How do you deal with repeated and correlated exposures? And we, at the moment, most of the work is focused on separate time periods, looking at separate time periods, and possibly where we think there's an important correlation, we adjust for that. But that's as far as we've gone with these models.
We did one last question.
Great. So I was wondering if psychosocial risk factors in pregnant women are or can be considered as part of the exposome.
They should be. In Helix, we didn't have very good data on, well, we have some social variables. We know the mother's education and we have some idea, we have a social economic status, we have something called a family affluence score. It's more generally about how well the family or the mother is connected socially and how they're doing basically in terms of affluence. But it wasn't the focus of the study. It was funded by environmental call in the European framework. So you can't do everything in this with the same accuracies that you would like to. But there's a lot of people saying now we need to spend a lot more, we need to put a lot more emphasis also on the social and psychosocial factors. So we realize that this is not the strongest part of this study, and it should be done, and it's an important part of the exposome. So, thank you for that.
Yeah.
I'm transported very challenging. So, I mean, maybe it's stupid, but you showed this for smoking, no? When they related to omics factors. So you are focusing in an external cause, smoking, and then projecting what are the relations with the. I was thinking it would make sense to do it the other way around, because the number of I just show very clearly the number of the size of the exposome is huge and contributions, and it's very complex the relations. But then when you nail this down to the genomic, it becomes much smaller. Like there are not that many genes, so there are not that many biological pathways. So I can imagine something like saying for the whatever RAS pathway, signaling pathway, that will cluster many different exposome reasons in a single biological and in a few biological pathways. So then you may see relations between exposome reasons that are then when they are translated into what happened to humans, they are not that many things that you can say in a human.
So you would first that way you would first identify the pathway of interest or a specific
pathways of interest.
disease.
You have a number of pathways, there are not that many, and they map the causes, and then you can also map the diseases. So they may act as a collector of information, reducing the complexity of the exposure and the complexity of the disease.
Very.
I don't know. Yeah, maybe completely stupid.
We started with the first atlas was just everything by everything, but I think if we have a more directed approach, it could help us.
I was just thinking in trying to reduce the complexity.
Okay. Good. Great discussion and
Good.
great talk.
Thanks.
Thank you, Martine.